So this is the third and the last video of molecular genetics. In this video I will start by solving the questions, in which I will start by the question 14. It says polyploid results from. We have to know that what we mean by the poly. It means many. Okay, as you know that in human beings we have haploid, which it is haploid, which it is one set. Okay, and we have, for example, we have diploid, sorry, we have diploid, which it is two sets of chromosomes. Okay, while polyploid, it means many. It means it may be 3n or 4n, and this is results during when we have non-disjunction. Non-disjunction, it means failing to separate, or for the separation of the homologous chromosomes during meiosis 1. Okay, during the process of meiosis 1. So polyploidy results from the non-disjunction. Disjunction means separating. Non-disjunction means they are not separating. Okay, when the gametes are forming. So when the chromosomes are not separating from each other, okay, one of the gametes contains more than it is normal number of chromosome, and it will result either of 3n or 4n. Okay, and it will result in the for the polyploidy. Aneuploidy, it's like having one extra chromosome, as we have it in the Down syndrome. In Down syndrome people, we have 47 chromosomes. Infection with a virus, it's not related. Polyploidy, it's not resulted from the virus, and the RNA process is a different thing. So the answer for the question is A. Okay, so the answer for this question is A. And now we have questions 15 to 19, which it is matching question. We have these terms, we have to match with the following statement. Okay, so the first one is gel electrophoresis. It means in this process we have a gel, okay, and we need electric current. Okay, so it's like a gel like this. It contains many pores, and we will put our DNA here. Okay, and we apply electrical charge from negative to the positive. It's connected with the electric. Okay, so our DNA sample will move through this gel according to the size of the DNA, the DNA of the size of our DNA sample, and according to the pores of the gel, the DNA will be separated. Okay, it forms many bands. Okay, in different places, so we can know the size of that DNA sample that we have. You can watch the process of gel electrophoresis in the YouTube videos, and you can find many videos that we have it. Just know that it is used for separating DNA samples. Okay, on basing on their size. Restriction enzymes, we said that they are like molecular scissors, in which they are used for cutting DNA. Polymerase chain reaction, it is a process, okay, which it is used for amplification. Sorry, amplification of DNA. It means copying of DNA sample, but inside the lab by using a machine. We called it thermal cycler. Okay, this is the name of the machine, while the name of the process is polymerase chain reaction. Recombinant DNA. From this term, it's clear in which two DNAs are combined. Okay, recombinant DNA. It's a DNA from two sources, we can say, or maybe from more than two sources. And RNA processing, we explained it many times in the previous videos. You know what we mean by the RNA processing. So the first question that we have, so in the question 15, it says refer to us molecular scissors in which they are it is, we can say, restriction enzymes, which it is B, used to separate large molecules of DNA on the basis of their rate of movement through an agarose gel in an electric field. And this is A, it is gel electrophoresis process, as I explained it before. Taking DNA from two sources and combining them into one cell. Combining two sources of DNA, this is D. Okay, it's recombinant DNA. Could cure cystic fibrosis. Cystic fibrosis is a genetic disease in which it is caused by a recessive allele, and this may be in the future, it could be cured, okay, by using a recombinant DNA method. Okay, it means that those people in which they have a defect in that gene, it can be replaced with the normal gene. Okay, so it can be, it's not done still now. So the answer for this one, it is D. An automated technique by which a tiny piece of DNA can be rapidly amplified, or we can say it can be copied. Okay, and the answer for this question is PCR, which it is, or we can say, polymerase chain reaction. Okay, it is PCR. This is the abbreviation for the polymerase chain reaction. The last question that we have in this video, it is question the 20th question that we have. So this question says 97% of human genome, okay, does not code for anything, and it's known as a junk. Okay, the second one says codes for about 30,000 genes, the 97 percentage. Okay, codes for about 3 billion genes. It's not true. It's located in the mitochondria, and this is also not true. It's not located in the mitochondria. Most of the DNA is found all inside the nucleus. It has been cut up by restriction enzymes. We cannot cut such amount of DNA by the restriction enzymes. It needs so much time. And also, for your information, restriction enzymes are found in bacteria. We don't have restriction enzymes in human. Okay, so it's not E, it's not D. Codes for about 3 billion genes. We don't have such number of genes. We have 3 billion nucleotides. Okay, the DNA is made up of 3 billion nucleotides. It codes for about 30,000 genes. This is not true. Just you have to know that the remaining 3% of the human genome codes for nearly 30,000 genes. Some source says 22,000 genes. Okay, but the 97% of human genome does not code for anything, and it's known as a junk. This is the answer for the question. In another term, we can say 97% of human genome is regarded as introns, okay, and they are not coding, and they are known as a junk. So this is the last question for the molecular genetics. I hope all the questions are clear for you. So if you have any question, if you couldn't understand anything, please just write a comment below. And thank you so much for your listening. I hope it benefits you.